Generally all living organisms have same basic needs. They require macromolecules, such as carbohydrates, proteins and fats, and water and minerals for the growth and development. Methods to study the mineral requirements of plants. Asterisk. In 1860, Julius von Sachs, a prominent German botanist, demonstrated for the first time that plants could be grown to maturity in a defined nutrient solution in complete absence of soil. This technique of growing plants in a nutrient solution without soil is known as hydroponics. Asterisk. By this method, essential elements were identified and the deficiency symptoms discovered. Asterisk. Asterisk. Hydroponics has been successfully employed as a technique for the commercial production of vegetables such as tomato, seedless cucumber and lettuce. It must be emphasized that the nutrient solutions must be adequately aerated to obtain the optimum growth. Essential mineral elements. C H O N K S Ca Mg Fe P Cu Mn B Cl zinc Mo Ni. Beneficial elements Na C She Co. Criteria for essentiality. Mentioned below is the criteria for essentiality of an element. A. B. The element must be absolutely necessary for supporting normal growth and reproduction. In the absence of the element, the plants do not complete their life cycle or set the seeds. The element must be directly involved in the metabolism of the plant. C. The requirement of the element must be specific and not replaceable by another element. In other words, deficiency of any one element cannot be met by supplying some other element. D. According to the above criteria, only a few elements have been found to be exactly essential for plant growth and metabolism. These elements are further divided into two broad categories based on the quantitative requirements, i.e., macronutrients, and micronutrients. 1. Macronutrients are generally present in plant tissues in large amounts, in excess of 10 mol/kg of dry matter. The macronutrients include carbon, hydrogen, oxygen, nitrogen, phosphorus, sulfur, potassium, calcium and magnesium. Of these, carbon, hydrogen and oxygen are mainly obtained from CO2 and H2O, while the others are absorbed from the soil as mineral nutrition. 2. Micronutrients or trace elements. These are the essential elements which are required by plants in small amounts, less than 10 mol/kg of dry matter. Micronutrients are mostly involved in the functioning of enzymes. Since micronutrients are required in traces, they are also called trace elements. Micronutrients are 8 in number: iron, manganese, copper, molybdenum, zinc, boron, chlorine, nickel. B. On the basis of function, essential elements can also be grouped into 4 broad categories on the basis of the diverse functions. These categories are: 1. Structural elements. Some essential elements are components of biomolecules and hence are the structural elements of cells. They are carbon, hydrogen, oxygen and nitrogen. For example, I, C, H and O are components of cellulose and many other biomolecules. Nitrogen is a constituent of all amino acids, proteins, chlorophyll etc. 2. Components of energy-related compounds. Some essential elements become components of energy-related chemical compounds in organisms. For example, magnesium is a component of chlorophyll that takes part in conversion of light energy into chemical energy. Phosphorus is a component of ATP which functions as energy currency of the living systems. 3. Enzyme activators or inhibitors. Many essential elements are responsible for activating or inhibiting enzymes. For example, Mg2+ is an activator for both ribulose bisphosphate carboxylase oxygenase Rubisco, and phosphernyl pyruvate carboxylase pepcase, both of which are critical enzymes involved in carbon fixation during photosynthesis. Zinc 2+ is an activator of alcohol dehydrogenase and Mo is an activator of nitrogenase during the nitrogen metabolism. Mn2+ is involved in photolysis of water. 4. Maintaining osmotic potential. Most of the osmotic potential of cell sap is due to inorganic salts such as nitrate, Cl, K+, etc. Osmotic potential is required for water absorption and maintenance of cell turgidity. Metabolismofnitro